with soils and ecosystems that are continually being degraded, using less water, fewer energy and natural resources, and do all of this under difficult economic and political circumstances. First of all, we need to understand you never change things by fighting the existing reality. To change something, you have to build a new model that makes the existing model obsolete. Also, we cannot solve our problems with the same thinking we used when we created it. So this is a paradigm change in agriculture. The biologically enhanced agricultural management approaches soils from a systems perspective. This includes all practices that enhance or improve populations of beneficial microbes in the soil microbiome. These practices include no or reduced chemical inputs or disturbance from tillage. We're finding out now that our application of nitrogen burns the carbon that you have out of the soil, and it also reduces the nitrogen resources as well. You want to also have year-round living roots from full-time ground cover of cover crops. This is a system, again, that we're rebuilding. And you can do this by either reintroduction of beneficial soil microbiomes, either through implementation of livestock, using what we call an adaptive multi-paddock grazing method, or we're seeing that you can inoculate with beneficial microbes from a composting bioreactor similar to what my wife and I have built. As we view the world, we need to see that microorganisms have shaped almost every facet of development on this living planet for the last 4.2 billion years. They've done this by increasing or improving the efficiency of the capture, storage, and utilization of energy on this planet. They've also created different levels of biological organization to facilitate this flow of energy. When we look at what microbes have done, 80% of the history of this planet, the Earth, microbes, bacteria, have been instrumental in changing this planet. You can see bacteria came along about 4 billion years ago. And shortly after, anoxygenic photosynthesis, probably the most novel adaptation, is enabling these organisms to capture energy from the sun, turn it to chemical energy as a storage mechanism, and then be able to utilize it later. We also see nitrogen fixation about 3.2 billion years ago, instrumental in building the building blocks of life, the proteins, the amino acids. And without this, this system would not be where it is today. Oxygenic photosynthesis about 2.4 billion years ago. Viruses about 2.3 billion years ago. And viruses are instrumental as agents of change. A lot of the evolution that we see has been promulgated by the viruses. The buildup of the oxygen in the atmosphere to about 2.2 billion years ago to 1.6 billion years ago, we see fungi start to come on the scene. Another segment of life that has been very important in how soils have developed. Mitochondria about 1.45 billion years ago. Utilization of energy increased 18 times with this adaptation. Single cell animals, 750 million years ago. Land plants, 700 million years ago. Glomeromycota, the fungi that associate with these plants and help them in their growth, about 400 to 500 million years ago. We just recently got here. We're just a little blip here at the top, and we are enjoying all that has come before us. So microbes are the foundation of every organism and ecosystem on this planet. You as a human, your microbiome is comprised of bacteria, fungi, protozoa, and viruses. You have about 10 trillion human cells. You have about 90 trillion microbial cells, and you have 1 quadrillion resident viruses. You are a very small part human and mostly microorganisms. Gene count, you have about 23,000 human genes. Your gut microbiome alone has 22 million genes. So you're outnumbered in gene count as well. What do they do for us? They digest our food. They do nutrient generation, vitamin synthesis that we can't get in our diet. They do cell renewal. They control our appetites and cravings. They comprise what we thought was 60% of our immune system, more like 80% or more. They detoxify carcinogens, prevent allergies and skin diseases. They even turn on and off our genes that regulate brain development, anxiety, depression, autism, arthritis, and emotional behavior. So what happens when these microbial communities are disrupted through either administration of antibiotics, exposure to chemicals, or diet-related issues? As you can see, most all of these are autoimmune diseases, and they're closely related to the dysbiosis of the gut microbiome. So many of these diseases can be cured by restoring intestinal microbiome structure with fecal matter transplants from healthy donors. This is one example of diet-induced extinction. You put in mice on a low-fiber diet. After 4 generations, you can see the diversity in the stomach microbiota of the mouse decreases year after year. We thought, okay, just bring the fiber back. This top chart shows you once you bring or reintroduce the fiber, you're still losing diversity in this system because you've lost the functional community that you had before. If you bring back the fiber and fecal matter transfers from a healthy mouse, everything is back and working instantly. So we know our farming practices over the last 120 years have created a soil that has lost its biological functionality, decreasing the ability for the soil microbiome to supply necessary elemental nutrients to plants from the soil parent material and fixed nitrogen from our atmosphere. If microbiota transfers work in humans, can a simple inoculation of beneficial microbes help restore microbiome structure and function in agricultural soils? We'll first look at how nature built soils. On the Great Plains in North America, we had bison herds that roamed around. They were kept in tight herds by the wolves. They could only graze about 40% of the forage there, and they would be moved on. They would deposit their dung, their urine, their saliva. They would also trample the grass that's there, the remaining 60%. The insect community, the dung beetles, would roll the dung deposited by the bison into small balls and place it in the ground for the progeny. A lot of this would biodegrade and compost in place because you have the perfect temperature and the perfect water content. The worms would effectively break it down as well, creating an inoculation event as the bison swept across the prairies. Now, not everybody can have bison or cattle on their property, so it's important that we understand that in order to restore that microbiome, we need to come in with an extract from this compost. Looking at how the soil microbiome population promotes nutrient extraction, understanding why we need to bring that community back. At the very base of this are your bacteria. They are processing either the organic matter from plant or exudates directly from the plant to the bacteria, feeding the bacteria. The bacteria are extracting elemental nutrients from the soil parent material. They're fixing nitrogen. They are the grass of this system, and that from that point, they are grazed on by protozoa, nematodes, and arthropods. What we see in bacteria, they have a carbon to nitrogen ratio about 4 to 5 to 1. The next level, trophic level up has a carbon to nitrogen ratio of about 15 to 1. So when these protozoa graze on bacteria, to get their carbon complement, they only need about 4 or 5 bacteria, but they're going to have 3 too many nitrogens. So they excrete that nitrogen as ammonia. And each level that you go up, more excretion of ammonia. This is where the plants and the systems get the nitrogen. Basically, bacteria are fixing it, but it's the protozoa, it's the system, it's the cycling of these nutrients by these different trophic levels that supply the nitrogen. Also, when you look at how nature has set this up, from a bare soil parent material like a desert up to your old growth forest, there's a ladder of succession that goes on through this. You see an increase in productivity from 90 grams of dry biomass per square meter in your desert soils up to 2,200 grams of dry biomass per square meter in your conifers and old growth forest. Along that change in productivity, we also see a change from bacterial dominant systems to fungal dominant systems. Currently in our agri-ecosystems, we have a fungal to bacterial ratio of less than 1, about 0.01 to 0.1. What we grow well now are weeds. Where we need to be is a fungal to bacterial ratio about 1 to 1. This is where your late successional grasses and row crops do the best. You can see about 600 grams of dry biomass per square meter productivity. As I say, if you don't have cattle or bison, this is another way. This is what my wife Wei Chen and I developed. It's called the Johnson Su bioreactor. It's a very simple system. It's built on a pallet. You put about 6 holes in it. You can see we're putting pipes in the center. The pipes allow airflow to come up through the system, so it's always aerobic. It's very simple. You have we use landscape cloth. It's about a 5.2 ounce landscape cloth, and a wire remesh, 6x6 by number 10 gauge remesh. It's used for pouring concrete and sidewalks. And you build a cylinder, and you have the pipes in the center. The pipes are only there for about 24 hours after you fill it. At that point, you pull the pipes out, and you have airflow up under the pallet, up through those holes, allowing it to be aerobic. You only irrigate it about 1 minute a day. It's about 1.5 gallons, or about 7 or 8 liters of water a day. Here's a link here that shows you how to build it. It's a vermicomposting process. It stays at 70% moisture content, which is ideal for both worms and fungi. So we have a lot of fungal development in this pile, a lot of fungal spores. There's no turning to it. It's aerobic. Worms are added after the temperature goes below 80 degrees Fahrenheit, 27 degrees C. It takes 1 year to reach maturity. And this is one important thing, is that you do need to reach maturity on this for it to be really beneficial. As you go through the composting process, it exhausts the nutrients that are there. And in that situation, fungi will sporulate. So you have billions of fungal spores in this community and a lot of diversity. Follow the instructions closely on the first try. Here's the link here below again. No shortcuts. Do not let it freeze or dry out. If you do that, a lot of times once you lose the worm activity, you have to take it out and build it, start over. What we noticed through the composting process from 60 degrees C down to 15 was a change in the bacterial phyla. You can see the proteobacteria start very strong, end up almost non-existent in the end product. This is why we're saying you need to allow it to go through that maturation process. Proteobacteria, somewhat strong, fade away, and then come back strong at the end to be a major component. And Bacteroidetes, the red, almost non-existent in some cases in the process, but they become a prime component as well. This is what the bacterial community looks like at 4 weeks, the top 80%. You have about 23 species of bacteria. At 22 weeks, when most everybody else says their compost is ready, you have about 57 species. If you allow it to go the full year, you increase the diversity by 4 times. You have 99 species in that top 80%. So it's important to understand that diversity is key here. When we compare 2 compost samples that were made 10 years apart, with different materials, the ending structure is remarkably the same. And this is what we're finding in our research. As we look at this, how a compost matures from right to left here, or more mature compost, you see the whole community structure changes. These are 10 Johnson Su bioreactors from the US. There are 2 from Australia, and I have 2, and there's also 2 products from Australia that are commercial vermicomposting products that are quite popular with the farmers down there. They like to use them because they do help in increasing productivity. You see a striking resemblance in mine and the ones in Australia, when the others here made in the US were quite spread out. But as you look at the maturity, how these populations of fungi and bacteria change through this system, there are significant changes by the time you get to that 1 year end period. At first, when we got this compost, we were curious about a couple of things. Curious about the impact of fungal to bacterial ratio and how that affected carbon and nitrogen partitioning. This was a greenhouse test with chilies, and we looked at carbon and nitrogen partitioning. First of all, carbon, the root carbon, shoot carbon, and fruit carbon are the brown, green, and red portions of this pie chart. The gray is new soil carbon, the blue is respiration carbon, and the orange is new nitrogen, expressed as the amount of carbon that it takes to fix that nitrogen. As you look at the change from a bacterial dominant system with a fungal bacterial ratio of 0.04 up to a fungal dominant system with a fungal to bacterial ratio of 3.6, also we have increasing soil organic matter in these as we fix these mixtures for the experiment. You see a change, only 3% of the energy that plant captured actually went to the plant in a bacterial dominant system. The rest of it went to new soil carbon, respiration, or fixation of nitrogen. As you step up and go to, and start to increase the fungal mass, 17%, 21%, up to 56% of the energy that plant captured went to the soil. To the plant. With that 97% here going to the soil, that plant must be in mayday. There was no logical explanation why a plant would devote that much energy to improving the soil unless there was a reason. And so we see that reason as increasing the microbiome structure and functionality. In agriculture, we sit at about 11% efficient. If you get to a fungal bacterial ratio of 0.368, we see 56%. So we should be able to improve this system 5 times as far as the productivity. Understanding that agriculture, we irrigate it, we fertilize it, and yet we only produce about 600 grams of dry biomass average, when old growth forests can do 2,200 grams. In our beam transitional, we're seeing we can match 2,200 grams of dry biomass per square meter. In an advanced beam process, when you reach that higher fungal to bacterial ratio, you have the carbon in the system, and you've rebuilt those nitrogen reservoirs. We're seeing about 3,186 grams of dry biomass per square meter. This is one of our small plot trials, just to see what the biology can do. You see behind us, that's the soil we're working with. We work in a desert climate. We're about 4,000 feet in elevation. So we're a high desert, very sandy soils, very few nutrients in the soils. We started this experiment on this particular area because it didn't have any legacy issues, any chemicals ever being applied. We just wanted to see, what can this biology do? So initially we started out with a 450 pounds per acre, 504 kilograms per hectare inoculation rate. It's a dusting. This is not a nutrient addition to the system. This is a microbiome, a microbiology introduction to the system, bringing the biology back in. Our first winter cover crop was about 400 grams of dry biomass per square meter. This is our second winter cover crop. We did about 1,608 grams of dry biomass per square meter. Now that represents 504 kilograms of nitrogen per hectare fixed in this system with the biology. This is our third season on our small plots. No fertilizer, just biology. We were growing 2,190 grams of dry biomass per square meter. Also understand this is a winter cover crop. It's planted in late November here. It doesn't get over a couple inches high over the winter. The system wakes up in mid-February, and this is the growth from mid-February to the end of April. So in 10 weeks, we're seeing 2,190 grams of dry biomass per square meter with the right biology. And again, 654 kilograms of nitrogen per hectare had to be fixed in order to support this plant growth because it is not in the soil. As we look over the season on this bar chart, the yellow is the uninoculated and the green is your inoculated compost system. Oops. On an annual production, we saw 3,186 grams of dry biomass. That's 872 kilograms of nitrogen because we are growing these crops, cutting them, hauling them off, and replanting. So there's no biomass being put back into the system. This is all biomass that's been grown. So we see up to 3,186 grams of dry biomass by year 3 in this system with the inoculation. Now, cover crops do work, but it seems they work better once you have the correct microbiome structure in the soil. This was the change in the soil. Our starting soil is just sand, no A horizon. After the 3.5-year small plot study, we had about 7.6 centimeters of A horizon in the control and 17.78 centimeters in the compost treated. Looking at best way to apply this, on the left, you have an extract applied on top of the soil, and on the right, you have an extract injected into the furrow. Now, the extract we're applying at about 2.2 kilograms compost per hectare in about 75 liters of water. There's no brewing to this. It's an extract only. It's conveyed in just a couple of minutes. When you apply this, you have about 968 million bacteria per square meter and 40 to 50 million fungal spores per square meter. This is an image of what the extract looks like under a microscope at about 400 times amplification or magnification. You can see these spores here, and the little white dots are your bacteria. And this is what you're looking for as you make an extract. You can see the difference between topical and injected into the furrow. We had over twice the biomass in the plants that were inoculated in furrow compared to topical, in both our spring biomass and our fall biomass. You can see in furrow, the plant reached reproductive maturity, where topical, it didn't. So we definitely see that there's a need for seed and soil and extract contact. This is a farmer in Belgium. He actually coated the seed with Johnson seed compost, just a little slurry, and you can see he also observed the same thing, double the productivity of the seed that had been coated compared to his control. This is some field trials we did. This was year 1. On the left is without the compost, and on the right, we applied the compost. And this one we did about 400 pounds per acre application rate. This is before we understood how small of an amount you could use. But you can see, without, we only had 50 grams of dry biomass per square meter, with 250 grams of dry biomass. This is year 6. On the same plot, we're doing 881 grams of dry biomass per square meter. And this is about 296 kilograms of nitrogen per hectare. Year 7, we're looking at 1,141 grams of dry biomass per square meter, 387 kilograms of nitrogen per hectare. So we saw a 5 times increase over this period over this 7-year period. So what do these microbes do for your soils and your crops? Well, you can see already they do not fix a lot of nitrogen for you. We see a lot of symbiotic and free-living nitrogen-fixing bacteria. You see phosphorus solubilizers. Now, a lot of the phosphorus in your soils is not accessible to the plant because of the soil physical chemistry. And yet when you have the right microbes that can release this phosphorus, it's available to the plant. Now, they also oxidize elemental metals from the soil parent material, furnishing all the phosphorus and potassium for these plants. They do carbon and nitrogen cycling, which is another very important thing. This system is like a monetary system on carbon and nitrogen. There has to be a flow or a velocity of carbon and nitrogen cycling in this system, so it's made available to the plant. In your economic system, if you don't have a sufficient flow, it collapses, and we see the same thing in these soils. The microbes also do phytohormone production, producing hormones that increase plant growth. But the most important thing that we saw as you build this community back is quorum sensing. The quorum sensing is when a group of microbes get together and work collectively, and they can translate genes that they can't as an individual. It's like us in a society. Individually, we can't accomplish much, but once we get into a community and start working collaboratively, then things started happening. And we saw that the quorum sensing, the activity was 6 to 10 times higher in gene expression than just regular housekeeping genes. So it's significant impact that quorum sensing has on this whole system. In the first 20 months of that study in the field, we saw an increase in micronutrient availability. Manganese and iron 1100% increase in soluble manganese and iron availability. Magnesium, calcium, zinc, and copper, about 40 to 83%. In this system, nitrogen, phosphorus, and potassium even increased in soils that we added none of this. So you can see in our soils, iron, manganese, copper, and zinc should be decreasing in availability due to the soil physical chemistry. But yet with the right biology, all of those are increasing up to 1,000%. You also need to see that as you add more carbon to the soil, you're going to increase the water infiltration rate, being able to accept water, and storage and availability of that water. That first increase in soil carbon percent will increase 5 times your ability to store water and have it available to the plant. So we get much better water use efficiency as you rebuild these soils with the carbon. When we went to field scale, what we did were cover crops between the commodity crop rotations, and we would plant directly into the row covers. This is Jay Young in Greeley County, Kansas. He completely cut back his nitrogen and phosphorus and came in with an extract from the Johnson Sioux compost bioreactor, and he's doing 200 bushel corn, or 12.5 metric tons per hectare. He saved $173,000 in 2021 by going with the compost extract and planting his corn. You can see the difference here in the seeds with compost and without. They're developing that rhizosphere. We have a project with Stella McCartney, a cotton project in Turkey. We're doing the same thing. We're coming in, we're planting cover crops, inoculating in furrow as we plant. We're rolling those cover crops, and then we're planting cotton. Normally, they would have 20 trips across the field for planting, fertilizer, herbicide. This is their schedule of what they have to do. We've reduced that to 4 trips just for planting. He did some insecticide spraying, so he had a 56% reduction in that. He had a total reduction in herbicide application. So he used 61% less diesel. He captured 18.94 tons of CO2 per hectare per year in the top 45 centimeters of soil. He also increased the soil nitrogen reserves in this 3-year period by 421.74 kilograms of nitrogen per hectare per year. He used 85% less fertilizer with no yield impact. His cotton yields were about 4.2 metric tons. The crops did well despite government-restricted irrigation and low rainfall. He only had 2 irrigation events on this one. No herbicides were used, and he observed biodiversity in the soil that they had not seen since their childhood. This is Corey Miller in Montana. He's using compost on a large scale. He's regenerating 1,000 acres. I'm going to try to see if this video will play here.
Last year we did a test we planted in the fall, and it was right when we were getting into the compost extract, and was it going to be a benefit? So we did a test where we planted a strip with our compost extract in furrow of cereal rye. And the original thought was that it probably wasn't going to do much. And now we're seeing the results of that. So we planted an entire 40 acres without it and just did 1 pass with our ash no-till drill, putting compost extract in furrow. And this year the results are remarkable. We have a plant, that's 8 inches was planted the exact same day, exact same time in soil right next to plants that we planted with compost extract. And we've got 4.5, 5 foot of growth on our extract plants, and we've got 8 to 10 inches of growth on our non-extract plants. To me, that was kind of the game changer. It's saying you don't put a seed in the ground unless you've got biology with it, because that biology helps that seed get established, and when that seed's established, and you got biology down there converting nutrients, that plant's gonna grow, to its maximum potential. This method of getting compost extract onto the fields is not expensive. We build our Johnson Sioux for less than $50. We have the materials on the ranch to do it. It just takes a little time. Getting that liquid produced is invaluable. You know, you can't put a price on it because it does so much for your soil. And when you start developing that and you start seeing the results, the success that you're going to have on your farm is significant. And we're seeing that. And we've done it in 1 year.
And I would say this link here is, he has some of the best video on the application of this as well, how he's doing it. And leave it to a farmer to be able to say more in less than in 2 minutes than I can in an hour. We're also looking at Ian and Diane Haggerty. They are pioneers on this. They currently do about 20,000 hectares, and they make an extract out of a very similar compost. It's a vermicompost.
They're making ours.
And they're also now making ours as well. They do barley in a dryland situation, and they apply an extract both on the seed at planting and then a foliar application at about 4 weeks. And they're able to get 3 tons of barley in a region that conventionally gets 1.5 to 1.6 tons per hectare. So they are over double the conventional production there. As you can see, the rhizosphere development on the seeds are just barely germinated. On the left-hand side, you see they've already harvested the barley. They got a late fall rain, and in nice neat rows where they had injected the compost extract was a C4 grass that they hadn't seen in 4 decades in that area. So this also reawakens the soil seed bank and helps it recover and come back with the diversity in the system. This is Russell Hedrick in Hickory, North Carolina. Excuse me. Doing dry land corn production. He averages 11.6 metric tons per hectare of corn, compared to the county average of 6.9 metric tons per hectare. He's using full-time cover, cows, sheep, and pigs. He's using animals to restore the diversity of the microbiome. He's got an 80% reduction in fertilizer application and no fungicides or pesticides in the last 6 years. In 2015, they had an exceptional drought, only got about 4 inches of rain. The county average was 0.5 metric tons per hectare. He did 8 metric tons under the same water difficulties. So there were a lot of farmers were coming to ask him how he was accomplishing this. So animals can bring this system back as well. These are kind of some of the more unusual observations we've had with this compost extract. This is Paul Transfield and Victoria. On the left-hand side, well, he sprayed this field with the extract, but he could only get on one side of the tree because this low branch on the right-hand side. This part of the tree leafed out a full month ahead of the rest of the tree. And the chestnuts off of it were twice the size of the ones on the other side. The Johnson Sioux compost with Rick Kay in Puma Springs in Healdsburg, California, Johnson Sioux, compared to an organic compost. You can see quite a bit of difference in growth. Alicia Taft, Rockfront Ranch, California, with her quince, with the Johnson Sioux compost, and this is what her neighbors look like without. And also Rod Rance and Rachel Ryan with hemp. They were getting 4 to 5 pounds of flower bud weight per plant when normal is 1.5, 1 to 1.5. And grazing wise, this is Alejandro Carrillo. Grazing, bringing back our grazing lands is another impact that this can have, having the animals properly grazed on the soil. Alejandro lives in the Chihuahua Desert here in Mexico, gets about 7 to 8 inches of rain per year, about 250 acres to support 1 cow. With AMP grazing, or what we call an adaptive multi-paddock grazing management, this is what he's done in a decade. He's changed the desert back to the grasslands it was. This is what we degraded it to. Originally, it was grasslands, and with our overgrazing of cattle, we brought in cattle, we brought in sheep, then we brought in goats, and it desertified the area to just all we have is creosote bush and mesquite. But with the right grazing, he's brought the grasslands back. This shows that these soils had a 4-foot deep soil carbon profile, So the grasslands had a significant impact on pulling carbon out of the atmosphere and placing it in the soil. We're seeing AMP grazing systems in the southeast U.S. when they're grazed like the bison were moved on the Great Plains. You can have over 2 times the carrying capacity when you properly graze these systems. We saw a 46% increase in forage biomass, an 18% lower respiration rate, and soil organic carbon at 21% higher rate. Now, I know you're talking a lot about carbon in Euro, and we're only looking at soil organic carbon. But when we start considering the whole system, the increase in forage biomass, the reduction in respiration, if we were to practice AMP grazing on the 1.25 billion hectares of savannas and grasslands, there'll be 18.8 billion tons of CO2 drawdown. That's about half of our current fossil fuel emissions. I want to caveat this carbon dioxide issue. CO2 is the second most important molecule on this planet. If we do not have CO2, it is the main component in the capture of energy, the storage of energy, and the utilization of that energy later. That's all the energy that we're enjoying right now has come initially from this molecule being processed in photosynthesis. The potential CO2 offsets on your grazing lands, as I said, was about 18.82 gigatons of CO2. If we were to enjoy or employ beam on our 1.36 billion hectares of farmland, there's potential offsets here from extra forage production, reduced soil respiration, increased soil carbon of 57.6 gigatons. Total potential is 76.4 gigatons of CO2. That's twice the emissions of the fossil fuel emissions on this planet. So the challenges have been accomplished. We can regenerate degraded soils and ecosystems. We can produce more food with fewer inputs, improve system photosynthetic capacity, and grow more crops per drop of water, using fewer energy and natural resources, while abating significant amounts of atmospheric CO2, and do all this under difficult economic and political circumstances. I thank you very much for the opportunity to present this research. And the ending message is align yourself with nature. She has a lot to be able to teach us on how we can best farm and how we can restore these soils. Thank you.
Thank you, David. That was a heck of a presentation. I think we can all say your results are just amazing. Hard to take in, really, the opportunities you're presenting there.
Farmers, yeah, I've got to give it to the farmers that are doing this and showing it the practicality of it. I mean, as an academic researcher, you can put this out and they'll say, no way. But once you see a farmer do this, like Corey or Jay, you know, this kind of brings it home that we can do this, we can fix this.
It has such potential because, I mean, where you were working with soils that were literally desert. Can I ask a stupid question? When you put those microbes in, what do they live off though? Like in the real desertification environments, do they not need to be fed again, or explain that cycle to me?
Once you get them in and get them in with a living root, see that's the other, this is, as I say, is a system.
Okay.
That makes this work. They're not magic. My wife will say yes on that one. It's not magic. It's just restoring the functionality of that system. There has to be the energy flow. That's always having a cover crop or having a living plant.
And that's why we often tell people you have to think about this as a living system. And within the system, that those microbes, they're like a very skilled startup. You go into the soil, you have the plants that they're giving those exudates to feed them. And in turn, those microbes giving back by getting those micronutrients that plants, they cannot get on their own. So it is a kind of symbiotic synergy going on there and go into the positive feedback loop.
That's what I was going to bring up. There's a positive feedback loop. You just keep cycling. You know, and a little bit of exudate goes back into the soil to feed the microbes. They're fixing more nitrogen. They're extracting more elemental nutrients from the soil parent material. They're recycling that back to the plant to pick up, increasing photosynthetic capacity. As you can see, year by year, we were increasing that photosynthetic capacity, increasing the flow of exudates back into the soil. As I say, it's a positive feedback loop that just year after year, this improves.
And one example, often the farmers will ask that, well, where would they get the nitrogen? Well, here in our atmosphere, we are literally pickled in almost 80% of nitrogen in our atmosphere. And if you have a healthy microbial community in the soil, they are bacteria, they can fix the nitrogen in the form that the plants can take up as a nutrient. So again, this is a system of the community, of the living system, that they work together to help each other.
Okay. Sean, can I hand it over to you? You want to comment on the presentation?
Hi, David. Hi, Chun. It's been brilliant. The figures just are starting to blow me away here. But I have a quick question. Just looking at the different operations you illustrated where you've used the compost on, the substrate for that compost, was it based on what was available in the farm? Like, for example, is if you had a mainly arable situation or a livestock, did you use that compost from the materials that came on that farm?
Yes, you know, any waste, moldy hay, stall clean out, leaves. That one slide I showed of the two composts made a decade apart, the original one was made out of dairy manure and leaves and yard waste, and the latest one was just straight alfalfa or lucerne, moldy hay. And it's not so much what you put in, it's the process that you put it through. And I think it's the conditions that the bioreactor allows that make the product the same. You know, to see, you know, that other chart I showed about how similar compost in Australia to the US, I've never seen anything so similar in all the research I've done in the metagenome. To have them all stacked on top of each other, they have different climatic conditions. They're making out of eucalyptus leaves there, completely different starting material that we have no access to here. And yet they're strikingly similar.
Thanks.
Okay. We have a question there that came in online. Leslie Dwyer, what is the best method to inoculate soil with the Johnson Soup extract in permanent grassland? What would you do in permanent grassland?
Well, we're seeing what Corey was doing. He was spraying it directly onto the soil. He was also coming in with a coulter and a drip system where the coulter would cut into the soil and it would drip into that furrow.
Okay.
That video that he did shows the best way to manage applying this extract that I've seen.
Yeah, we truly appreciate the innovative farmers. They see a resource as a tool, and then they will innovate a way to utilize it. And as long as you have the living roots out there, the injection or the spraying will work. But if you are planting and with just seeds, then you will need to have a ways to inject the extract, the compost extract, while you're planting the seed, and make sure there is seed contact with the extract, because you need the microbes to help those seeds when they germinate.
Well, they actually help them germinate as well.
Okay,
So
because you had coated some seed in it as well. Wasn't that one of the examples?
Yeah.
Yeah.
Yeah, that's also another method, is to make a slurry out of the Johnson Soup compost, and then you will use the slurry to coat the seeds. And the amount that you use is very little. For example, you only use 1 quart of that slurry for like 50-pound bag of seeds. So that is very small
It's about 4
inoculation
stone
rate.
bag, isn't it?
Yeah.
Okay. Here's another chap about the grassland. We're applying animal slurry to grassland instead of chemical fertilizer. Can we add the Johnson soup compost directly to the slurry in the tank, and would that help?
Here's the problem with the slurry, and you do not have the biology to accept and process that much material. I taught toxicology, and we always found out it's the amount that determines the toxicity. You know, in our life, if there's not enough of something, we have issues. If you have the right amount, things are working good. If you have too much, it's toxic. And if you don't have the biology there to break it down, I'm not sure, this would have to be, there's a lot that we don't know on this. You could try that, adding the biology to the system, but it's having it into the soil as well. Applying these slurries are an issue, and I think there's so much of that, the nutrients are so mobile, the nitrogen, the phosphorus, that they do just kind of leach out or go into your waterways. I think isn't that one of the issues you have in Europe?
Yes, it
is the excess nitrogen.
Yeah.
It seems like it would be best to pre-process that and get it locked up into an organic structure so it's not so mobile. See, that's what we're seeing. The nitrogen that we're rebuilding in the soils is locked into an organic structure. So it's not soluble, it's not leachable, it's not available until you have the microbes break that product down and make it available to the plant in that process that I talked about. So it's.
I try to make a more visualized form that people can have. Is, for example, you won't go out there and then grab a handful of wheat and eat them that way. You have to have some skilled people, the farmers or the millers. They mill the wheat and then make them into flour, and you still don't eat the flour by handful. Then you have some skilled people to make noodles out of them, to make yummy baked goods out of them that we have hard time denying them. And that is a process making things that's edible, digestible, and able to absorb. So same way in the way of certain nutrients out there in the nature is that the plants need to have the microbes as skilled experts to process all these different nutrients and then make them into a form that the plants are able to take up and able to absorb and being a helpful and useful nutrient.
And there are processes to stabilize those nutrients in that substrate. So.
Okay, we have a question here. Are you doing any sap testing of the plants to see how much of the macro and micro mineral increase in the soil is being taken up by the plants?
Well, no, I'm not doing any, but what we are noticing, the cows in that one from Australia where he sprayed it on the pasture, the cows went where he sprayed it.
Okay.
And I trust a cow more than I trust a test.
Yeah, they're choosy. Well, if it's good.
They are.
Yeah, they are. Jim, do you have questions? Jim is an organic grower. He's doing a presentation as well, one of Ireland's best sort of organic growers, and he also teaches a lot of courses. What's your take on this process?
I'm just like a child in in a Whole Food shop, we'll say. Thank you, David. So my question is, so your container, how many acres will it do?
Okay, about 2,000 pounds of material, wet material, will go into the bioreactor. By the time it's mature, we see about 700 pounds. So it'd be about 350 acres that it would cover at 2 pounds per acre rate. So it's a considerable amount of area for one bioreactor.
Yeah, go on.
And also, this is not really a question, it's an observation, David. I'm familiar with your work, even though I've never done it, and your YouTube videos. A number of my students have taken it on, but they have not followed your instructions to the T, and it has failed. It's an observation, but I appreciate that at the very beginning you said you must follow the instructions carefully.
That's for my wife.
Yes, because this is a 100% aerobic composting system. Also incorporates the worms in there, and they are the red wigglers, the composting worms. And you're keeping at a 70% moisture content, which is ideal for the worms to thrive, and also for the fungi as well. And so there are certain protocol that you need to adhere to of building this. The reason why we designed it in such a way is because the material that we use, for example, the woven landscaping cloth, they are tight enough, but yet has enough porosity that air infiltration is appropriate, that keeping this system aerobic, because air can infiltrate enough. But also it has enough density that it does not allow too much evaporation of moisture, so you keep the moisture content constant in this system as well. And that is very important because just like us living in a structure, a shelter, we like to have certain comfort to thrive. And so if our house is leaky and it does not keep warm, then it gets too cold, it's hard for us to survive as well. And this is the same thing for those microbes. Just have to think about that this system that you're building is a microbial incubator. And you're trying to make sure that they live through this system and then they mature to the way that microbial structure is going to be beneficial when you bring them onto your land to inoculate your land. And that's why it takes a whole year. That's a maturity, maturation period that it needs to adhere to. This is what I often tell the farmers. It doesn't matter if you keep your wife in a very warm room or feed her extra food. It still takes her 9 months to have the baby mature to the right maturity and to give birth for a happy, healthy baby. So we get this question a lot. Too often people ask that, what if we heat up the bioreactor? Well, it doesn't, we don't know what it would affect, but we know that it's not going to make it mature faster.
Okay. Another question there from Claire. This is coming from our online audience. Can the system work if there's lots of one particular input? For example, different farming systems with a lot of animal waste or waste from dairy or lots of green material. We touched on, I suppose, slurries there, but what about a lot of green material, and what was the other point you made? Green material, does it work with that? Or a lot of animal waste? Well, same as probably manure, maybe. Manure is better than slurry, right?
Yes. Well, most of the material we deal with here is dry when we process, and we do run it through a chipper shredder. I did dairy manure at the very start of this. Now you need to keep the density of pile up so it's not too dense. So I did add wood chips in when I did the dairy manure, but for the most part, the wood chips will not break down in that year. So you're losing that much production of the compost. Compost ends up to be like clay. You can squeeze it between your fingers and it'll push out like a ribbon. And as far as, you do have to follow the instructions as much as you can and let it mature. And you can see that I'm able to track with the metagenome. It's too expensive right now to be able to do everybody's compost with that particular test, but we're working on a less expensive test that can be done cheaper and faster. So you'll be able to judge the maturity. There is a certain point that you see there's a complete shift in the dynamic. You saw in that metagenome chart that I showed you, a complete shift in the dynamic of this. There's a complete reversal in fungal to bacterial ratios in it, which was odd because the finished compost shows a bacterial community dominance compared to the fungi when you just do the counts.
But as far as the mass goes, the fungal mass is still dominant.
Yeah, you have to consider a fungal spore. One fungal spore can house about
Yes,
2,000
it's much
bacteria. Yeah, yeah.
I'm just going to check questions from the floor. Okay, there's two here. We go to the bearded gentleman first.
How does your work relate to biodynamic farm practice in the preparation of their sprays and so on?
We're not familiar with their process as far as how exactly it's made, but we seems to get quite a few people think that they're similar.
Yeah, we've been wanting to test the biodynamics, get a good sample from somebody to see, is this the same thing? It does seem like the same thing as far as what the results that you see, as far as biodynamics and their increase in productivity.
But we don't have any volunteers yet for their for sample.
Yeah, we'll get you
We'll
some
get you
here. Okay, next question.
You mentioned the figure of 500 kgs of compost per acre or per hectare. That seems very low to me. And the other thing, when I put FYM or compost on a field, I put out maybe 2 tons per acre. And the other thing is, you mentioned in Australia 10,000 acre farms, and presumably you put compost on those farms. How do you put compost on the 10,000 acre farm? Or I mean, how do you supply enough compost for a 10,000 acre farm? It seems that to me that your system is more suited to a small horticulture situation.
In contrary, because that particular farm in Australia, they are making their own compost with our method now, and what they did was that they used the compost to make compost extract, and then use the liquid to inject when they plant their barley or wheat. And then also, like what David said during the presentation, they also come back about in 3 weeks or a month and then do the foliar spray as well. And they have set up a system that is a larger batching, but adhere to all the principles and all the steps of the instruction. And they also designed a 20,000 liter extractor to make their compost extract. So that's why I said earlier that it is so amazing to watch some of these innovative farmers, that if there are something they think it will be beneficial for their operation, and they will innovate to utilize it. And that's exactly what happens.
So you can scale it up and it still works really, really well, like in that massive scale.
Yeah,
Yes.
2 kilograms per hectare. I mean, as I say, there's no nutrient value in this. It's the quality, the inequality of the compost. I mean, there's plenty of compost that some of them are detrimental when you put them on the soil because of the salt content. Go ahead.
I just want to go, one question here and then back to Sean.
David, yes, very good presentation, very impressive. The question I'd ask, am I right in saying the process is far more important than the feed material, and you're not that concerned about stuff, fertilizer growing, some chemical being in the process as long as the process is right?
Well, I don't know that you need the fertilizers.
Yes, I get that, yeah.
If we can cut back 85%, and this was in a transition, they cut back 85%, and they're able to match conventional production. So eventually you can get off of fertilizers. And I think that's going to be your best path forward because the damage that these fertilizers do. We don't realize how much carbon they burn out of that system. And carbon is just as important as that nitrogen. It's the energy resource for those organisms to function. You know, the biological functionality depends on energy, just like our society depends on energy. You take energy away, as you can see, the whole system collapses. You know, production collapses. You know, creature comfort, food supplies. You know, it's the same thing in the soil. That if you don't have that carbon flow, like the carbon cycling in the system, the system's broken. It's not functioning the way it should. You're not able to grow what you need to grow.
Okay, Sean, next to you.
Yeah, I'm just wondering, if you go with, we'll say, one of the challenges we have here, we've fairly damp climate, so we're trying to, if we want to avoid using glyphosate or Roundup, and do less soil disturbance from a tillage operation or an arable. Have you seen your results? You mentioned weed suppression. One of our challenges is that the crops, we get weed before we get the crop germinated, if you know what I mean. Mm-hmm. If you have any comments on that.
Well, in Turkey, they had the same issue. They had 3 or 4 runs of herbicide. Before. But once you brought in those covers, roll those covers down, those covers keep the weed germination at bay. And they allow, when you plant that, whatever commodity crop, either cotton, corn, chili, I know your crops are much different over there, but if you can plant into that cover, then you get so many benefits. You're building up a detritosphere where that cover that you roll down is feeding the next crop. It's feeding the microorganisms that you need. It supplies the resources for the bacteria, the fungi. All of those systems start to work again. And if you notice, most of the rangeland, you know, it had a surface cover of detritus. And if you look at how soil is built up, it's really built up layer by layer. You know, the carbon that we're trying to measure now that is in the soil profiles is a little different. But if you look at nature and you look at the carbon dating on the soils by depth as you come up, the oldest carbon is at the bottom. The youngest carbon is at the top. You are rebuilding your soils. You know, many of, as I say, we're losing our soils, but they can be rebuilt year after year by employing these covers, employing the biology, increasing that photosynthetic capacity in this system. And planting into these covers seems to be the best way to do this.
And we noticed that both in Turkey and also another farmer in United States, that as they try this method and coupled with the cover crop and with a no-till system, was that they were able to reduce or actually totally do away with herbicide, because the weed suppression, the roll down cover crop, and also that the change of microbial community in the soil change that structure of the soil that is not as friendly to the weeds anymore.
Mm-hmm. Okay, Jim, you had a question?
So, it's almost the same question. So in a vegetable garden setting, what is your observation around weed growth and your compost?
Well, in vegetable, I thatch or mulch everything. That keeps I don't deal with weeds, because with a significant mulch on there, the weeds can't get through it.
Yeah.
So that's kind of the key. In order to rebuild these soils, you need that cover, either living or thatch, to feed that soil system. And that's where we kind of messed up in agriculture. Is, you know, we're pulling everything off. You know, we cut the wheat, get the seed, and then come back and take all the straw. And there's nothing there to feed that system. And you continually will be fighting this. Whereas if you come in and start, just like what we see in grazing, leave 60%, harvest 40%. This is the kind of a golden ratio. If you look at corn, the corn seed is 40% the biomass on that field. The forage is the other 60%. Wheat, cotton, chili. All of these run around 40%. That's what nature allows you to take. You need to have that other 60% go back and feed this system to rebuild it. It takes that much energy for this system to be able to survive and increase its fertility.
Yeah, as they rebuild and regenerate, and it will recover faster for you. So again, we go back to that positive feedback loop. And just like on the pasture, on the grazing land, if you don't take too much, that those forage, those plants are able to regenerate better. And then too often that they are able to capitalize on the moisture allow, or the rainfall come down, and then you're able to bring the livestock back quicker. So there is a balance as a manager of your system, need to observe and then adapt and pivot according to what you see on your land.
Okay, we'll take one online question and then we'll have to wrap up, even though it's so interesting. We're just running late.
We'd like to keep going too.
What is the best way to apply the extract onto grass by foliar means? Do rates vary for different times of the year?
Foliar, yes, spraying. If that's one way. I kind of like what Corey did, getting those coulters and cutting a groove and dropping it into the furrow to get it into the soil. But yeah, as long as it's a living cover, yeah, I think you can get by with spraying. We haven't done that research, but we've had farmers and ranchers that have tried it and been successful in it. As you can see, Paul Transville. Another thing he noticed on that, when he sprayed his fields, he always went out to harvest the cow patties for his garden. He noticed when he went out after spraying, the cow patties were gone.
So he had enough microbial community, including the dung beetle species, that you basically incorporate and then process.
Yes.
The dung.
Yeah.
And so to that question, the answer to that question, perhaps that you need to, if spraying is the easiest method, then you can deploy that because you already have the living roots. And also try to spray during the season that there will be growth as well, because that's when your microbial community in the soil can be most active to work with your plants, because that's when the plants are doing the photosynthesis and capture that photosynthetic capacity. And also that if you can time it before a rain event, probably not a hard thing for Ireland, but you got to look at it is that you need the rain to allow those microbes to go into the soil as well.
Amount-wise depends on the how good your compost is. You could try some double application, triple applications, and see what responds best on the compost that you have.
Okay. We're going to have to say goodbye. Your talk was absolutely wonderful. And did you want to say something?
I just wanted to say to David and brilliant. I just think it's fascinating. And we're hoping that we might see you in person in Irish farms, hopefully in the spring.
Well, we're hoping to.
Yeah. So we'll be chatting about that in the next few weeks. So thanks a million.
Okay.
So everyone say thanks to David and Stephen.
Thank you.
Thank you, guys. We're going to have a tiny short break and come back to Jim. Poor Jim, you've been waiting. But thank you, everyone, and thanks so much.